Yeah, I think they must be the writers. Yeah, and push falls over the room in anticipation. This is good news. Okay, I think we're ready to get going here. So our speaker here is so famous that she's infamous. To steal a line from a movie. This is Angelique Stephanou, who's going to be talking this morning on cancer metabolism. Of course, one of the key cancer hallmarks and one that's of considerable interest to at least one of the groups here today. So it is my absolute delight to turn this over to my friend and collaborator and person I just adore as a scientist and as a person, Angelique Stephanou, who will talk about tumor metabolism. Angelique, the floor is yours. When we get down to about a 5-minute warning, I will do a little react emotion emoji thing and ping you in chat, and then we'll have plenty of time for Q&A afterwards as well. Thank you, Angelique. Looking forward to your talk.
Okay, thank you very much, Paul, for the very nice introductions. And I want to thank all the organizers for inviting me to give this talk. So I'm very sorry I could not come here to be present with you. So I had the last minute problem, but well, I'm here. So I will be going to talk about metabolism. So we've heard a bit about it already. So I will discuss this through, and we will see how it goes. So it's essentially a work that I've been doing with my 2 PhD students, Pierre Jacquet and Ala Tafek. So they are both graduated now. They both have their PhD diploma, and so they are postdocs in other institutes. So let's see. Yes. So you have seen that many times already, the hallmarks of cancer. And the thing is, I'm going to talk especially on one particular one, which is the deregulated metabolism. So when we hear about deregulated metabolism, we understand that something is abnormal. So the whole question of this talk will be to discuss this point. Is the tumor cell metabolism really abnormal? So there is a big increase in interest for the cell metabolism. So we've seen many papers published over the last 10 years about these particular topics of tumor metabolism. And so I just made a quick search using the keywords metabolism and cancer using PubMed, and I had lots of papers coming around, and I just put the abstracts and make the cloud of words just to have a feeling of what's coming out of these 2 keywords. And what we see at first is that the therapy looks like a really important thing with the targeting, the resistance things, and the treatment. So there is a huge therapeutic importance around this metabolism, and the key issue is to search for a metabolic target. So when we search for a metabolic target, it means that we want to find the difference with the normal situation, so that we can target precisely what is not right with the cancer. So this is a very active subject, the search for the metabolic target. And also there is a lot of drug repurposing. And since there are already plenty of drugs existing to treat metabolic disease, and in particular, there is one disease which is diabetes 2, the type 2 diabetes. And for this specific treatment, so for patients who have this pathology and cancer, we discovered that the cancer was developing slower than for all other patients. And the question was, why this particularity? And it occurred that these people were taking a molecule which is called metformin to regulate the glucolysis. And then the metformin is a kind of slowing down the metabolism, and then it has a huge impact, in fact, on the treatment of cancer. So there's something interesting here that we can look around and to see if we can not couple some drugs specifically targeting the metabolism and standard drugs for cancer treatment. So if we look deeper into this, what we see is like we have lots of keywords coming out, like glucose, the reactive oxygen species, stress, hypoxia, oxygen, and all is around more or less hypoxia and glucose, of course, which are the two substrates for the metabolism. And what this is stressing is that there is a dysregulated environment around the tumor. And so the question is, most of the time we consider that it is the cell that is dysregulated, but is it really? Is it not more interesting to look specifically into the environment that will stress the adaptation of the cell? So that would be the whole question of this talk. So for a start, I will recall what I want to describe specifically in the metabolism. So the metabolism is hugely complex. So I'm as a modeler, I will make a crude simplification of this all problematics. So what I see, what I understand, and what I will describe in this in my model is very simple things. Is that the metabolism is based on the use of glucose, and then it will come into glycolysis to transform the glucose into pyruvate. And depending on the environmental condition, especially the oxygen amount, then we will go into two different directions. The first one is respiration, when there is sufficient amount of oxygen. So this is promoted, it means that the pyruvate will be transformed into acetyl-CoA using the pyruvate dehydrogenase. And this enzyme will transform into this substrate, and all this reaction will occur in the mitochondria. At the opposite, when there's no oxygen, then the pyruvate will be transformed into lactate using the lactate dehydrogenase, and this occurs mostly when there's no oxygen. So these two enzymes are key in these talks, because I will focus on the expression of these two enzymes as a signature of the respiration and fermentation. So there is one key point here that I want to stress, is that most of the time what we hear is the opposition between OXPHOS and glycolysis. But in fact, we need glycolysis to produce OXPHOS. It's the first step before OXPHOS. And in any case, we have to transform glucose into pyruvate, and it's pyruvate which is directed into two different ways. So we hear most of the time glycolysis in opposition to OXPHOS, but in fact, it's just the first step of OXPHOS. But I will say it in my talk, because most people do, and I do the mistake either. So we will oppose glycolysis and OXPHOS as two different ways. I will try as much as I can to use respiration and fermentation, not to make the confusion. And then, in fact, fermentation is oxygen or anaerobic glycolysis, which is the correct term, which is to add anaerobic in front of glycolysis. Okay, so I hope this is more clear. So the standard way is to consider the metabolism normal when we just have this dichotomy in between the two ways of metabolism. When in presence of oxygen, when we will go into respiration, and in the absence of oxygen, we will go to fermentation, which is the normal way that cells behave. And in the tumor case, what we observed, it's an observation that was made by Otto Warburg a long time ago. He started his observation in the 20s, and he wrote his very nice paper in 56. So he has the Nobel Prize for his research on the tumor metabolism. And what he observed is that when we have a culture of cancer cells, we observe a huge amount of lactate produced in the medium. And in his idea, he thought that the mitochondria might be defective in tumor cells, and it was his main hypothesis to explain why the tumor cells were producing so much lactate. And this was independent on the amount of local oxygen, whether there is oxygen or not. There was always more lactate for tumor cells than for normal cells. So which activates the LDH enzyme. So if we go back to the paper of Warburg, it is, however, very interesting to notice that he makes no hypothesis about the level of oxygen in the medium. Here I stated it is in the presence or absence of oxygen, but he didn't make this difference. For him, aerobic fermentation is voluntarily not discussed. So it's too labile and dependent to external conditions. So he make no hypothesis about the level, this environment. But now what we see in the literature, it's always that the Warburg effect is always associated to aerobic glycolysis. So it means that it's just when the cells are in presence of oxygen, the cancer cells, they just produce, they just use the glycolysis, which is the fermentation, as their main mode of functioning. So we will see if this is a real will to describe it this way or not. So in the following, I will distinguish between the Warburg original, which makes no assumptions on the level of oxygen, and the modern definitions, which I call the modern Warburg modern, to specify that we need to have oxygen to have the glycolytic mode. So we can understand why this new definition was used, because in the normal case, when we have oxygen, then we expect the cell to go back to oxphos. So we understand why there was the specific stress to say that even when oxygen is available, then we have a change of metabolism. So it's legitimate, I think, to use this modern definition. Okay. So it's interesting to see that in most cases in the literature, we see that there are normal cells with their normal metabolism that switch in between fermentation and respirations. And then when they become cancer cells, then there is some kind of switch that put them into a Warburg phenotype, which means that they will be producing, using fermentation even in presence of oxygen. So is there really a switch, which is a binary mode in between these two, or is it an oversimplifications? So this is the old questions. Is it discrete or continuous? So the other option would be to consider that there is not such switch, so defined switch, but something much more continuous, with a continuous adaptation with all the gradients from in between the oxphos and the respiration and the fermentation, which can be understood by the fact that the level of expressions are all evolving with time, always evolving with time, and there's no such threshold as would suggest a switch. So it might be more continuous. Okay. So we also hear a lot about reprogramming. So if we look at the amount of publications about the cell metabolism, we see on the yellow curve that initially it was the switch term that was mostly used to describe the transition between the normal state to the tumor state regarding metabolism. And then suddenly the switch term was replaced by the term reprogramming. So is it more appropriate? With my student, we wrote a paper on these questions, because we think that it's a very fashion. It's a fashion to use terms that comes from computing to make biology more understandable, more tractable, and maybe it's not true to the true sense of biology, which is maybe not that deterministic and might be more not chaotic, but with more fluctuation and heterogeneity. So the question of, is it the right term to use reprogramming to transform normal metabolism into tumor metabolism, is something that I will address too. So what we understand when we use the term reprogramming is the idea that there is some irreversible metabolic alterations, and that are mostly due to mutations. So what we are going to see is that if we really require some mutations to see some diverging metabolism, or can it be just emerging? So another key points when we consider metabolism is the acidity, because the glycolytic, the fermentation, is producing a lot of lactate, and together with the proton ions, which means a lot of acidity in the environment. So it's a well-known fact that the tumors are very acidic. So, and when we again read the literature, we often hear about the inverted pH gradient in cancer cells. So is it true to state it this way. So in normal cells, what we mean is that in a healthy normal tissue, the extracellular pH is around the physiological level, which is around 7.4. And inside, if we measure the intracellular pH inside the cell, then we observe it's slightly lower, around 7.2. So we have an intracellular pH, which is smaller than the extracellular pH. And in cancer cell, what we observed is, of course, the extracellular pH is much more acidic because of the hypoxia producing the fermentation and then the acidity. So we are around 6.2, even sometime lower. And inside the cell, measurements have been made, and mostly what we found is that the pH seems to be much higher inside the cell. So that's why we see in the literature the fact that there is an inverted gradient in the cancer cell. But in fact, it's not a property of the cancer cell to have this inverted gradient. It's just a reaction to the environment. So what is triggering the change of the gradient is just that the fact that the extracellular acidity is decreasing. So that's why the cell wants to defend itself from these acidic aggressions, and then it will activate a number of proton pumps to just expulse the protons, so that it keep an intracellular pH, which allows the cells to keep functioning. So this is important too, because the way things are said have a lot of importance on what we can understand of these facts. Okay. So as a curiosity, with my PhD student, we have the opportunity to make some experiments to measure the intracellular pH in some cancer cells. So it's a very simple experiment. We just need to use a microscope, fluorescence microscopy, and use a fluorescence probe to just measure the pH. So as it is simple, we decided to just make this experiment and see how the acidity is regulated inside different types of tumor cells. So we focused on two cell types, human glioma and rat glioma, so U87 and F98. And we just measured the intracellular pH by submitting all these cells under the form of 2D culture and under the form of spheroids. So here is the typical shape that tumor that we had. So it's about 200 microns of radius. And so we put these cells into a plate with a 96 well, so we can test many different conditions. So we vary in each well the extracellular pH, and we varied it from 5 to 8.4. So most people say why it's such a large range of pH, because it's unexpected to find something as low as 5 in vivo and as high as 8.4 or 0.4 in vivo also. But since we are in experiments, then we can test whatever we like to see how to push the cells in its where it never goes. So each measure was performed 3 times, and you just make this measurement. So just a few words to explain how the methodology to do that. So as I said briefly, we use the spectral method, which used a molecule which is called BCECF, which is a pH-dependent probe, which means that the fluorescent spectrum is depending on the pH. So here we have different culture medium with different pH, so we can see it in the color, which is slightly changing, and specifically the emitted spectrum. When we excite it with a laser, then we see different shape, different intensity, and this gives the information on the pH. So what we did is that here it's difficult to distinguish in between the different pH. So the methodology is to use two peaks typically. So we need to have 2 lasers to excite the probe with 2 different lasers, so that we have 2 peaks. And the common methodology is to make the ratio in between the two measured peaks, so that we have a kind of normalizations, and we get rid of the amount of probe inside the culture medium. So it's a very nice way to measure it, but it means that we have to excite twice the sample to have these two peaks. And the methodology is for this BCSCF probe is well known with a given laser that we didn't have on our microscope. So we were a bit annoyed by this. So we decided, well, we'll do with what laser we have. So we tried it with 4 different lasers, and so to measure the different peaks. And we observed some signals, which is a pH dependence with different things. So we decided, why not try all the combinations to see if we can improve the accuracy using different lasers. So this is what we did. We excited with all the available laser on the microscope, and then we made all the ratio measurements, and we had the ground truth by measuring effectively the pH with a pH meter in the culture medium. And what we found is that the best ratio that we can find was with these two peaks. So we had to excite the solution with these 2 lasers, you can see 409 nanometers and 458 nanometers, and then we make the ratio in between the 2 emission peaks, this big one and this small one, and then we get the best accuracy in our measurements. So that was fun to do. So it was apart from the normal modeling job that we are doing, but it took us some time, about a year, to make all the measurements and to derive the right methodology. But it was fun to do, and we were happy with that, especially because we were able to significantly increase the measurements, and also the range in which we could use this specific probe, BCSCF. The theoretical range was to measure pH between 6.8 and 7.4, and with our methodology, we can significantly extend the measurement that we could make from 4 to 8. So that was great, because it's exactly what we wanted why we wanted to submit our cells to extreme pH. Okay, so the good point is that finally we get a relationship in between the ratio measured from the spectral method and the measurement of the pH. So it means we have to excite our samples, and then we get the ratio, and then from the ratio, then we can measure the pH. So it's a very simple measurement. And this is the result we obtained with our 2 cell lines. So initially, what we see on this on these curves is the different pH in which they have been the cell have been immersed, from 5 to 8.4. So with the different color, so the red is more acidic and blue is more basic. And we measured the intracellular pH, so the probe can be internalized into the cell, so that we can measure not in the cell medium this time, but inside the cell, the intracellular pH. So we measured it. So initially, in the F98 cells, for example, the baseline pH, intracellular pH, was slightly above 7.5. And in the other line, it was 7.5 exactly. So initially, the intracellular pH was there. Then we immersed the cells into the different pH, and then we observed how the intracellular pH is evolving according to this. And we observed that after 1 hour for the 2 cell lines, they just take the extracellular pH. So it's too a violent process. So the cell is not able to react to this shock, and then they just equilibrate the pH external and internal. And with time, over 8 hours, we see slowly the intracellular pH that is going back to something more or less normal. So we see in the F98 cell line that the pH is not able to adapt and remain mostly very acidic. So the cell is not able to expulse the intracellular protons. So it remains very, very acidic. Whereas in the other cell line, it was nice to see that the cell could significantly expel the protons and could rise their internal pH and adapt, regulate this pH in a way. So the first thing which is interesting is to see that the behavior is different regarding the cell line. So it will be interesting to measure it for a much larger amount of cancer cells to see how they behave regarding the acidity. And this is a very important signature of the metabolism, intracellular metabolism. So what we have is in the first cell line, we have a high resistance to acidity. I'm not presenting the result, but if we make some viability test on these two cell lines, we observe that the F98 is able to survive very acidic pH, lower than 5. And whereas the other cell line, the U87, is not able to survive this very acidic pH, and the cell, they mostly die. But they have this regulation ability so that they can preserve their viability. So there was a real question is by which process can they resist such high acidity? So we made some very preliminary observations, and it seems that they are encapsulating the proton ions into some liposomes, some small capsules, and it neutralizes the acidity intracellularly, and so that the cell can keep functioning. But it's very preliminary work. We don't have the ground truth for that yet. So it's interesting to see that in these two cell lines, we have two different behavior. So mostly the F98 have an intracellular pH which tends to be lower than the extracellular pH, whereas it is the opposite for the other cell line. And so the pH gradient is not always inverted. So it's not a common point for all cancer cell lines. It can vary enormously. So we just take the two cell lines, and there are so many others to test to see how it behaves in other lines. Okay, so now the question about cell metabolism is the Warburg effect really a hallmark of the cancer cell? So we published a paper about that, about searching for the metabolic signature of cancer, if we could find it. So it's a review paper to try to have a landscape of the literature on these aspects. And what we found is the first important thing is that not all cancer cells are glycolytic. I mean, they use fermentation. For example, under lactic acidosis, the cells tend to revert to a non-glycolytic phenotype. And this is called the reverse Warburg effect. And in fact, it is the reverse Warburg. The Warburg is the production of lactate, and the reverse Warburg is the fact that the cell, they tend to import the lactate inside intracellularly, and then the lactate is transformed into pyruvates, and this will fuel the OXPHOS and reactivate OXPHOS through the mitochondria. So the acidic environment is a driver to this, seems to be the driver to this process. Another interesting point is that the Warburg effects exist also in normal cells. So what we see typically is that when a cancer cell is reactivating its OXPHOS, it's producing some reactive oxygen species that can create some oxidative stress upon the neighboring cells and normal cells. And for example, in that case, in cancer-associated fibroblasts, this was observed. So it activates the IF pathway. So IF is the hypoxia-inducible factor. And this will trigger the pathway to glycolysis that will produce a lactate. And this is interesting because there will be some kind of symbiosis in between the two cells. So one will produce a lactate that will be exported inside the extracellular medium and will be imported by the cancer cells that will fuel their OXPHOS. So it's an interesting mechanism where we have a cooperation in between cancer cells and tumor cells, and normal cells and tumor cells. So tumor metabolism, the whole question is it abnormal? So we have a bunch of questions to solve. Is there a switch? Is there really a reprogramming? So is the metabolism is discrete? Is it discrete binary, or is it more continuous in the sense that it is more adaptive? And the whole thing is Warburg really a hallmark of cancer? So to try to understand all these questions, of course, we developed a model. And so we started by trying to develop our own model, but it took us a long time to decipher all the literature, to make the assumptions and everything. It's a hard task to tackle the metabolism. There are many existing models, but they are too huge to be tractable. So it's a difficult task, but we found a very interesting paper that was more or less considering all our constraints that we pre-established. And this paper was published in 2020 and by a Chinese team. And it was studying the metabolism at the scale of a single cell. And it was quite simple because it was combining some genetic regulations together with the two main metabolic pathway, fermentation and respiration. So we can find them here, the two chains of metabolic spots. And then this is connected to the gene regulations, which was really important for us to answer our question about the effect of adaptations and also about the reprogramming. And so it was producing this ATP. So this was the starting point for our model. And then this team had a very nice way to represent the evolution of the metabolism using the two enzymes I mentioned before, which are the PDH and LDH. So they propose a metabolic landscape in which we can see how the cell evolve in this landscape. So if the PDH is expressed, it means that we are more in a oxphos mode, in a respiration mode along the vertical axis. And if we move along the horizontal axis, then we go more into the overexpression of the LDH enzyme, which means we are in a more fermentative metabolism. So it's a very nice landscape that shows that we can see all the evolution of the cells through this landscape. So they noticed in their studies that there were existing some specific attractors given their conditions that they expressed in their model. So they identified mainly 4 attractors, one for normal, one for more glycolytic, and an intermediate state, which was very interesting, and a proliferative one, which requires a lot of oxygen to boost the proliferation in their conditions. So, okay, so that was first, I would like to say an important point about this model is that it was at the cell level. So it was a metabolism model to describe the metabolism for just one single cell. But in our case, we want to see how this metabolism evolved in a growing tumor. So we integrate this into a hybrid multiscale model. So yeah, we've heard a lot already in this workshop about this type of model. And I thank Paul for the very nice presentation yesterday about the PhysiCell, because finally it's the software we choose to use for this task. So we tried many different softwares and implementations. So we had our own implementation originally, which was to use a cellular automaton, but we were very quickly limited with these representations, because in the cellular automaton, basically we have 1 cell per voxel, and which we could not take into account the changing density of the cells in the medium. So it was huge limitations, because the increased density is very important so that we can generate the proper gradient of oxygen in the environment. So we have to move into some kind of lattice-free model so that we can take into account this density. And so PhysiCell was absolutely brilliant, because we could do that. Everything was here to be used. And so to describe it in more details, so we have basically 3 different scales. So we have the environmental scale, where we will describe the substrates that are diffusing around our tumor. So I will describe them just after. And then we have the tissue scales, which describe the tumor growth. And for each individual cells, we can plug our metabolic network, our metabolic models. So that was done for 1 single cells in the previous paper. And then we solve these equations, these models for each single cells in our model. So just to mention that it's very timely in computational cost, because we wanted to have things as more accurate, as more realistic as possible. So we used a huge amount of cells. So we had the grid, it was about 200 by 200 by 200 since it's a 3D model. So it means that we reached 1 million of cells. So our simulations were taking about a week in a huge computer grid, calculation grid. And it took us about a week. But most of the simulation I will present, except one, which is really a huge simulation, we did it in 2D, so to reduce the computational cost. But we checked that in 3D, we observed the same kind of behavior. So here it is. So of course you had all the details of the PhysiCell model yesterday, so I won't go into the detail, but we take into account the cell cycle and proliferation. So we can take into account the mutations, and in the genetic networks we have the degradation, consumptions of the substrates, production, secretion, and the whole metabolism that we described. And the key point to run the model is to take care of the time. Also, Paul mentioned it yesterday. So we have different time scales. So the reaction kinetics are very fast, the diffusion also, and whereas the growth for the tumor takes about hours and days. So to look at the environmental scale, what we did is to solve a bunch of PDEs. So we reduced the number of variables that we take into account. So we took into account only the two main substrates, which are oxygen and glucose, and of course the lactate, which is produced by the glycolysis, and the related proton concentration, so that we are able to calculate the pH in the environment. So it's standard PDEs, which for each some diffusion, and for also some discrete terms that represent individually the consumptions of each cells in the environment, depending on their locations and their collaborations. So mainly for oxygen and glucose, we have depletion terms, whereas lactate and protons are mostly produced. Okay. So, as I said, we use this model developed by the Chinese team, but we had to adapt it to our specific problematic. So the original model has 56 equations, 119 parameters, 3 matrix parameters, matrix of parameters by quite huge matrices, 56 by 56, and everything was calibrated on experimental data. But we didn't do that. It was done in the original paper, very nicely done, with lots of research on to find the experimental data to do the job. So it was really nicely done, and we had very few parameters to estimate by ourselves. So the thing that we have been changing is that, as I said, we have the reverse Warburg effect that can occur, that was not taken into account because the environment was mostly static in the original study. So we took this into account by adding the regulation by the environmental pH. Also, we introduced the lactic acid, the lactate that was able to come in and out of the cells. In the other original paper, it was also static. And also the role of oxygen, which was quite crudely taken into account in the original paper. So we just make some improvement on this so that we have a better relationship associated to the production of ATP. So this is basically the main modification we have to do on the original paper. And so it's also some other adaptation that could be made, and that we have made. It's to look specifically at the regulation induced by the enzymes. So we use some yield functions to define the expression of this enzyme, and we have a parameter gamma, which is used to decide whether we have an inhibition or an activation, which is a down regulation or an up regulation of the genes. So it's all decided by the value of this parameter gamma that we introduced. So in this case here, for example, if we have an inhibition, then gamma is smaller than 1, and if it's bigger than 1, then we have an up regulations, and if it's 1, it's a normal behavior of the gene. Okay, and so we have a truncate the curve so that we do not have a behavior that is not corresponding to the normal behavior, like up or down. So this was some adjustments. So this was nice because it gives a way to act on the regulation and on the activation of the genes into our model and see the relative importance of the gene in this metabolism. Also, for all the reaction speeds between the different substrates, we used very standard things, which are Michaelis-Menten functions, which are very standard in this type of model, in metabolic models. So I won't describe it further. It's very standard procedure. And the good point is that we have the saturation of the curve. And here how we proceed. So in the original models, the Li and Wang, what they did is that they used a lot of simulations. So they initially each bead, yellow bead, is a cell. So initially they start with a random initialization of the intracellular parameters for all the cells. So they have lots of different cells, lots of different cell identities. And then the extracellular concentrations are kept constant. And what they see is the metabolic landscape is emerging from the huge amount of cells with different parameters they are putting into the simulations. So they have to realize a high number of simulations for one given cell so that they can cover the whole landscape. So the landscape is emerging through the amount of simulation they are producing. And we have a different strategy in our case, because here what we have is an evolving environment. So the extracellular concentrations, they will evolve. So what we did is to start with all the cells quite similar, with similar parameters, with partially random initialization of intracellular parameters. So they are very close by in terms of identity. And then the extracellular concentration will evolve as the tumor will grow. It will change the environment. It will shape the environment, and the metabolic landscape will emerge from this unique simulation in our case. So the landscape will be, we will see how the cell will go around the metabolic landscape during the growth of the tumor. So what we will represent is the individually each cell in this metabolic landscape, and we will have the density map of these cells in the landscape. So as I said, so it's a very well-known thing. When a tumor is growing, then it progressively depleting the oxygen inside the heart of the tumor. So the white circle is the boundary of the tumor. And initially, we have this amount, a certain amount of oxygen, a certain amount of glucose, lactate, and the pH, they are all close to physiological level in vivo conditions. They were chosen that way to represent an in vivo situation. And then the oxygen is depleted as the tumor is growing. And of course, the glucose also is depleted. And at the opposite, the lactate is increasing because of it's a byproduct of glycolysis, and the pH is becoming more and more acidic. So as a consequence, if we represent the different variables, the different substrate proteins taken into account in the metabolic network, here we represented two highly known, which are if the hypoxia-inducible factor. And we see that the hypoxia-inducible factor in presence of oxygen, it is degraded. And when there is not enough oxygen, then it can be expressed. So essentially, it is expressed at the heart of the tumor. And it's the same for VEGF, since if we directly activate the production of VEGF, and VEGF is the vascular endothelial growth factors, which will stimulate the angiogenesis, so that the cell becomes vascularized and can get some more oxygen to keep growing. And then the important point in our case is to trace the LDH and PDH levels. And so the LDH it's an indicator, is a signature of the glycolysis fermentation, sorry. It's the indicator of signature of fermentation, and the PDH, the signature of oxphos. And here what we see is we have a high level, obviously, a high level of LDH in the heart of the tumor and a small level of PDH. But interestingly, what we see is that we have some pockets of cells that highly express the PDH all around this tumor. Okay, so now it's nice because we have a model that allows us to really to measure the different amount of the enzyme produced, so that we can map exactly the different metabolic modes. So we make some gradients because it's not discrete. In fact, it's much more continuous. But we classify the different type of metabolism into 4 cases. One, it is fermentations mainly with a little bit of respiration, which is purple. The red one is purely fermentations, and we use the pink violet one, which is respiration with little fermentation, and the respiration only. And what we see, we have all these different classes of metabolisms that can be traced inside the growing tumor. And with time, so we follow the evolution of this metabolism. So we have this that is organized into different layers that are quite expected, with the more glycolytic in the center and the more the respiration at the periphery. And as the tumor is growing, then we see that we have a massive necrotic core occurring because there's no more substrate able to sustain the proliferation of the cell in the center. We also observe some holes coming in when the necrotic core is completely depleted. And it's interesting because we had this in our experiments, and it was fun to find it also occurring in the simulations. So this is the basic metabolism when the glucose goes into pyruvate, and we have these 2 modes emerging. And now if we look specifically at the Warburg phenotype, so this is where I make the distinction in between the modern Warburg and the original Warburg. So in the original case, it's just lactate excreted independently on the level of oxygen. And in the modern definition, we have the aerobic fermentation, especially at the periphery. And what is interesting to observe is that we observe some reverse Warburg, which is where the lactate is imported inside the cell. And this corresponds exactly to the previous picture that we saw here, where the PDH was overexpressed at some points in yellow here. So it is strongly associated to the reverse Warburg, in fact, this OXPHOS metabolism. Okay, so I check on the time. Yeah. So now I represent the metabolism in all for all the cells. So this is I represent for each individual cell their metabolism, and I map it into the landscape. This is what we obtain for the different time. So initially we have some OXPHOS, and then with time we move into the more fermentation mode on the horizontal axis. And then after 20 hours, we see that we come back to OXPHOS, which is respiration, so which is due to the reverse Warburg in part, and this is even more striking later on at 30 hours and 50 hours. And then as time passes, what we see is that we have a overexpression of this kind of intermediate states, which is neither in OXPHOS fermentation or respiration. It is somewhere in between because it's a dominating state. So this is interesting because that shows that there is no kind of dichotomy in between OXPHOS and respiration and fermentation, but we can be somewhere in between. So this is what we get. This is a summary of the observation for the metabolic landscape. So we have a massive oxidative state expressed, a glycolytic fermentation state also, and this interesting intermediate state. And the neutral state that was mentioned in the original study was not observed. It comes from the fact that they were using some random parameters. So they were using parameters that were out of the normal ranges. That's why they get this kind of neutral state that we cannot find in our specific model. Okay, so this was the summary of the metabolism, but the whole point of the model is that we have a very powerful tool to try to make some experiments, numerical experiments, and see what happens if we change the conditions, the environmental conditions, and play with that and see what we can get. So this is exactly what we did. So as a first test, what we did is we changed the level of oxygen in the environment. So we produced some cycle of hypoxia with varying the duration of the cycles. So 18, we tested here we tested many different frequencies of hypoxic cycles, but I retain 2 here. Since they are quite different. So when the changing oxygen cycles are 18 hours, what we see is that we maintain a pool of respiration. So cells maintain this metabolic mode during the tumor growth. Whereas when the hypoxic cycles are much more extreme, much more bigger, then this metabolic mode is completely disappearing. Okay, another test that we did is to make some acid shock, because as I explained earlier, is that when we put the cell into acidic conditions, then we can modify the metabolism, especially we can have this reverse Warburg effect. So that was interesting to test. So initially the pH we kept the pH with a physiological condition homogeneously into the system. And what we observe is that we have absolutely no respiration at all, or very few cells going into this mode. Whereas when we increase the acidity of the environment, then we get this oxphos coming back. So the respiration the metabolic mode is reactivated. And interestingly, if we observe also the number of cells that express the Warburg phenotype in this case, is that when the environment is at the physiological pH level, then we have no reverse Warburg at all. And but the original Warburg is also of course expressed a lot. But here we have this reverse Warburg, which is directly due to this new metabolic mode of reactivation of oxphos. Finally, the last test that we did is to deprive the environment from glucose and see what happened, but it was pretty obvious that we completely eradicate the glycolysis, the fermentative mode, and we just have the respiration one. Okay. So to sum up, what we did is that we played with the environment, and with this kind of way to do things, we could explain how we can go from one trajectory to another, we can go from one state to another. For example, if we increase the lactate, then we move from the oxidative state to the glycolytic state. If we decrease the pH, then we go back to the intermediate state and go back to the oxidative state. And glucose also, when it is increased, then we can reinduce the glycolytic mode. So it's a nice way to have the different transition in between the states in the landscape. So we did another simulation. So this one was done in 3D. So it took a long time to produce it. It took about a week. And in this case, we wanted to force the cells to give it plenty of oxygen. So it's the level of oxygen is much higher than in the simulation before. The level of glucose is smaller. And we remove completely the lactate in the environment. And we started with an initial pH, which is slightly acidic. So that to push the cell not to use to be far away from the Warburg phenotype, to be pushed to use oxphos and see what happens. And so what we observed is that we change a bit it changes a bit the shape of the cells, because the metabolic modes were changing more drastically. So we have a more irregular shape for the tumor growth state. which was the first interesting point. And also in the environment, what we observed is, if we plot here the acidic environment, we observed in this particular simulation, but it's not common to all the simulation, it's just in this particular one, some kind of dipole forming with two sources of lactate that produces also acidity. And interestingly, we have on the border some kind of lactate diffusing food from one pole to another. And the consequence is the following. We see it on the right picture, blue and orange, that we have some local phenomena, local Warburg occurring just on this particular pole, and all the other regions that were fueled with the lactate coming from one pole to the other, they express the reversed Warburg phenotype. So that was quite interesting. And the essential point of this simulation is to show that, in fact, the Warburg effect is really a local effect. It is reversible because it's changing with time as the tumor is growing. And it's most of the time, it's a transitory phenomena. So it's not something that we can consider like a constant expression of the cancer cell. It's very something which is emerging from the environmental conditions and which is evolving with time and which is really dependent on the conditions. So we have no need for a reprogramming to express the Warburg phenotype. We don't need to induce and to call for some mutations or overexpression of genes in the systems. Just spontaneously, we see it emerging and evolving with the conditions. So the last thing that we did is we had to play with the gene regulatory network I just talked about to see what was his influence in the trajectories. So if we plot the standard metabolic landscape corresponding to the last simulations, we observe these three attractors, which are basically the oxphos, the intermediate, and the fermentative profile. And if we change the expression of the genes, especially the gene which is influencing lactate, which is influencing the expression in the systems. So if we change just the parameter gamma to make it more sharp, what we see is that we just deepen the attractors into one specific location, one specific point. So it's just that we focus more the behavior. So instead of, we reduced, in fact, the heterogeneity of the system with the mutation. So the mutation is really a stronger attractor than the normal expression of the genes. Okay, so we'll arrive to some conclusions. So the cell metabolism, that the main point of my talk is that it expresses a great diversity, and it is strongly dependent on the environmental conditions. And the Warburg effect emerges spontaneously, so we don't need any specific mutation to see it occur. And finally, this is the whole question we were asking through this talk, is the tumor metabolism is not fundamentally different from that of a normal cell metabolism. It's just the environmental conditions that are really different, with the acidity and the hypoxia first and the acidity second, that drive the metabolic state of the cell. So this is interesting because in therapy we really need to address, but there is an increased awareness of this, that the environment is very important, and we need to take into account the evolution of the environment and consider the whole system as an ecosystem to have the cell and its environment. It cannot be considered independently and alone. And okay, so I just added this slide because of the conversation on the opening talk. Thomas Hill, and he just mentioned the atavistic theory of cancer, and it is a subject on which I'm quite interested. And I just, with a colleague, we just produced a review paper on the subject, and we were asking the question, is cancer metabolism and atavism? And as you explained, Thomas, that indeed the atavistic theory of cancer proposed that the evolution of cancer is reverting the evolution of life. So it means that the cancer is going back into an ancestral state in which the conditions were harsh, that there was no oxygen, a lot of radiations, so the cell has to survive independently and behave as an individual, not as a member of a collectivity, like they lose their social behavior to become selfish and look for substrates, for nutrients on their own. So there's no cooperation anymore. So it recapitulates, in fact, the ideas of this atavistic theory of cancer is that the evolution of cancer is going backward the evolutions. So the cancer cell is losing its ability to behave with as a multicellular entity and goes back to the origin where the oxygen is lacking and where the acidity is high. And so we discussed this particular concept. Oh, sorry. And we find our conclusions. So I don't want to spoil the paper for those who want to read it. But our idea is that we cannot simply, it is not as simple as that. Because in this evolution of life, if we agree with the atavistic theory of cancer, then the first thing that should change in the cancer cell, since it's going backwards, would be the last thing that would change would be the metabolism, because metabolism is a very, very early event in the evolution. And in cancer, the problem is that we see it emerging very early in cancer development. So from this point, it cannot recapitulate the evolution of life, since the metabolism is altered first and not late as it should be if we were following the atavistic theory. So this is our conclusion of the paper. But the paper, the original paper that was published by Davis, Linweather, and also Mark Vincent, who I met in the fields, in fact, a few years ago. And we talked about the atavistic theory when I was in Toronto, so it was a few years ago. And he was very a pioneer in this theory and discussed it a lot. And so now they published lots of papers, and there is an upgrade with a 2022 paper by Linweather, and where he described make some improvement on the original theory to explain how atavism can be understood not as a whole series of changes, but as a discrete steps in time that can be traced back following phylostratigraphy trees, and which is very interesting to follow. So the theory is really nicely expressed and mentioned, but still there are some small bits that need some improvements. Okay, so I don't know if I might be out of my time. So just to conclude about the origin of cancer, which is interesting, because the whole question is do we have to call for mutations, or is plasticity enough to express most of the cancer phenotypes? So in my talk, I was mostly caricatural because I was extreme in the fact that there was no mutation involved and so on. Of course, there are lots of mutations. There's no problem about that. It's just that when you start cancer development, do we really need it at first? Do they come first, or do they come later because of the deregulation of the environment? And this comes into the debate that is quite active. It has been active for some time about the two theories to explain the origin of cancer. The first is the somatic mutation theory, which say that it's just a mutations that created cancer, and then it develops because it acquires some abilities that normal cells don't have, and then it will deregulate the environment. And the opposite theory is the tissue organization field theory that suggests that there is a stress in the environment, and this stress will be the main driver for the changes in the environment that will finally trigger or favor the mutations, and then the mutation comes second, and they are not the first driver. So there are lots of discussion around these two theories, and this is quite interesting to follow. So to conclude, so what about the hallmarks of cancer? So I come back to the picture that we have seen many times in this workshop, and the question is, do we have a hierarchy in the different hallmarks? So which one is more essential than the other? Is it possible to decide this one is more specific to cancer or not? Or can we find an ordering of the different hallmarks. So which one come first? Because, for example, the induced excess, the vascular, the angiogenesis hallmark is just a consequence of the hypoxia, of the deregulated hypoxia, and then the deregulated metabolism that creates hypoxia, and then it will induce the vascularization. So some are depending on some others. So it would be nice to kind of find a hierarchy. So when we see the last publications, there are an increasing number of hallmarks, but maybe we should go backwards and try to find less hallmarks, and which ones are really fundamental, are really central, and which ones are more peripheral. That would be a nice way to see things. So I will stop here. So I just want to finish by acknowledging my collaborators. So this is a work that has been done, essentially what I presented by my 2 PhD students, Pierre-Jacques and Ala Tafek, and all my team. And also I have a strong collaboration starting now around this question with Mathilde Badoual, which is in University of Paris, and Jibin Poatil, that maybe was supposed to be in fields. I don't know if he's here or not. And so we are working actively also on these questions. So I thank you all for your attention, and I will be very pleased to answer any questions.